Great, thanks Ryan. Thanks everybody for being here today. It's a pleasure to speak with you this afternoon. My name is Matthew Kinn. I'm Senior Vice President of Business Development and Corporate Initiatives here at Recursion. So, as we've alluded to throughout the day, we have a three-prong approach to driving value here at Recursion. All good things come in threes. Of course, we have three of them. First is our internal pipeline, which is focused primarily on precision oncology and rare diseases. You heard about it this morning from Laura. You will hear more about it this afternoon as well. Second are broad research collaborations in complex therapeutic areas, such as our existing collaborations in neuroscience and undruggable oncology targets, as well as future collaborations that we might enter into in other intractable areas, such as cardiovascular metabolic disorders, inflammation, and immunology. Lastly, are partnerships and agreements related to data and models that underpin the Recursion OS. And you've heard about many of those models and data sets earlier today. Let's dive a little bit more into our partnerships. As a reminder, in December of 2021, we entered into a pioneering collaboration with Roche and Genentech. The goal of this collaboration is to identify and advance up to 40 therapeutic programs in the fields of neuroscience broadly, as well as a single oncology indication. Last year, we were very excited to announce that Genentech had exercised its option on our first validated hit series under the oncology arm of that collaboration, and we look forward to sharing additional potential future milestones related to both projects as well as map options here in the near term. So what's really exciting, I think, in addition to the progress that's been made to date, is actually the like-mindedness that we share with our Roche and Genentech counterparts. We're delighted today to be joined by John Marioni, who's going to speak a little bit more on this topic. In addition to having a distinguished academic career, John is the Senior Vice President and Head of Computational Sciences at Genentech. As well, John's a member of our Joint Governance Committee under the collaboration. So, John, I'll invite you up. Yeah, turn it over to you.
Thanks, Matt. So it's an absolute pleasure to be here. It's been a really fantastic day so far, learning about all the amazing work that goes on at Recursion, and seeing some of the examples and the demos is really cool. So I'm going to start with an old image. And this is just to say that this isn't new, what we're thinking about now. Technology has always driven our field. This is an image of a microscope from the 18th century. And Robert Hooke used it to look at slices of cork. And within that cork, he observed those structures. And that's the first use of the word cell, because it reminded him of the areas where monks used to sleep. So that's the etiology of the word cell, and the first time it was used as a microscope. But of course, we've been looking at cells as a field using various techniques for literally hundreds of years. And it's a reminder that as the technology evolves, the use of the microscope, the use of stainings, the use of single-cell genomics, it allows us to get more and more information about cells. So this is an image from some work of my own where we were looking at early development in the mouse context. And it's a reminder that technology, not just in the area of looking at cells, but in other areas, has really been a driver of biological discovery. It's been a driver of the work right across the field. And at the moment, we're on the cusp of another major technological leap forward, and that's artificial intelligence and machine learning and how we use that effectively. And that's going to be the next thing that's going to push this field forward, as we heard about so much this morning. But of course, it's not all solved, otherwise we wouldn't all be working on this, we wouldn't be thinking about this so much. We've got the ability now, in the labs that we can see to the left and all around here, to generate huge amounts of data from both healthy and perturbed conditions. But it's critically important that we make sense of these data, and in particular, can we predict perturbations that we've not seen? So in an ideal world, what we would like to be able to do is to predict in any cell type of our choosing what would be the impact of any perturbation, genetic or chemical, and we could then use that as the way to really motivate the experiments that we're going to do. This is not a trivial problem. There are tens of thousands of cell types, tens of thousands of genes. Once you start adding variants and think about different combinations of variants that act epistatically, this is an enormous search space. And compared to other areas of the sciences, so in physics or chemistry, the actual number of constraints is fairly limited here. We don't even know how to model fairly simple processes like transcription or the cell cycle. If we can't model that, then the ability to understand the impact of a particular perturbation in a complex multi-tissue organism is really tough. So we believe, and I think that this would be something that would be shared by everybody in this room here, that this is one setting where computational models, and especially foundation models and generative AI, can truly transform how we both discover and develop medicines. Now, as Najat just said, there's a number of different facets where these approaches are going to be applied. I'm going to focus on the discovery angle here. But of course, it's also true that you could use these approaches to design better molecules. You start with information about structure, affinity, other binding properties, etc. And you could use that to search through different areas of the chemical space. And later on, when we think about designing trials, absolutely, but also in automating that process. Regulatory filings are slow. It's not glamorous. It's not going to improve your PTS, but it is going to decrease the time it takes to get from that molecule that works to a filing that's going to impact the lives of patients, which is what we're all here to do. So within GRED, so that's Genentech Research and Early Development, I lead the computational sciences organization. It's through this that I have such a great understanding and a wonderful partnership with colleagues here in Recursion. And within Genentech, we're the computational part of this that's making this vision a reality, from everything from infrastructure, data management, through to model development and application, all the way through to phase 2 trials. How? Well, it's actually a very simple and similar model to the one that we've heard a lot about today. It's something we call lab in a loop, you call it virtuous cycle or virtuous circle, but the idea, the concept is very similar. You start with an experiment that's able to generate very large amounts of data, and I do think that it's important to emphasize that the scale of data generation is really critical here. We've previously gone with very targeted data generation. That ability to sample randomly from across the whole space is really important because we don't know what we don't know. There's large areas of chemical space, but especially of biological space, where we just don't know what this perturbation is going to do. So that ability to really generate data, perturbation data from across that whole space is really vital. And having generated that data, you then use that to feed those models that are then going to be able to make the predictions, that are able to stitch the data together, and then to generate those new hypotheses that I talked about, that ability to predict what is the impact of a particular molecule in a particular cellular context. What's going to be the impact of that genetic perturbation, and in time, how do they interact with one another? Just a very simple example to illustrate why that ability to generate data matters so much. Here's some strings of DNA. Very simple problem. We want to take an 80-mer sequence of this, and we want to be able to predict what's its effect going to be. How expressed is that 80-mer going to be in a random context? Well, the way that you might typically have approached this problem in the past would be to design 80-mers, perhaps based around known genes, around known RNAs that you could use in order to test this. But actually, that's limited. And so what this is work from my colleague, Aviv Regev, that she did, she decided that instead of actually trying to synthesize in a targeted way particular sequences, they would just generate 10 to the 8 random sequences from that 4 to the 80th space. And they were able to show, using this, that they were able to generate training data, where they were able to get how expressed each of these sequence tags were, and then build an Oracle-based model, just a relatively simple transformer, and using that, they were able to show that they could be able to predict the expression of an independent random 80-mer better than those models that were building upon known sequences, so the 10,000 or so synthesized sequences that people had been focusing on in the past. So really that combination of computational technology, 80-mer sequences that you could generate at random and at high throughput, and the ability to really work tightly between the different spaces, the wet lab, the dry lab, computational science collaborators on the underlying infrastructure, so important. But of course, that's a relatively simple problem. We want to scale this up to really discover new targets here. And we think about this across all aspects of R&D, where we go from the experiment to the data to the algorithm really quickly, and it's that tightness that matters so much. We've really got to have those readouts that are relatively simple to generate, and we've got to be able to generate them at massive scale, because we don't want to have a wonderful readout that takes 18 months to actually give you something that you could then feed back in. You've got to have the right readout that you can use at scale. That's going to enable you to generate those models that are then going to be useful. And of course, there's more to it than that. There's also these strategic partnerships. We share a partnership with NVIDIA, both Recursion and Genentech on the computing side. We, of course, have a great partnership together, and we have a variety of other partners in-house, including, I just want to emphasize, AWS here, with whom we work on quite a lot of the computing infrastructure also. Okay, so where do we hope this is going to get us? Well, we hope that it's going to get us, and I think this is a shared goal again, to that right target or chemical matter for the disease that you care about. So together with Recursion, we work with you guys in a couple of areas: gastrointestinal cancers, the first system, and then that very broad collaboration in the neuroscience space. And in each of these, starting with that cellular system, and I would just say that it is harder in the non-cancer space, it's something that's worth emphasizing, to have the right model system that you can use to actually generate the relevant data. That is just a challenge. But having generated that system, you can then target it with CRISPR, with known small molecule compounds, and you can see how it reacts. And you can measure that output in a number of different ways, for the cell painting over on the left, but also single cell transcriptomics. So perturb-seq-like assays, other assays that you could use to read out the impact of these perturbations. And having done that, you can build models. You can either build unimodal models that will consider one of the assays independently, so imaging and single cell RNA-seq, and then think about how to bring it together. Or you can build multimodal models, and those are really very powerful, where you're able to combine the different types of data together in a single model. We've heard already today about how that combination of data really matters. And then having done it, what we all want to be able to do ultimately is to be able to predict better targets, better compounds that are going to have the effect that we want in these cellular systems, bring that through into the clinic, test those, and fingers crossed, they read through positively and impact the patient's lives, which is what we really want. So just a little bit upon the model development, because there's a lot that's gone on in the sort of the data generation. We've heard a lot about that today, but of course, the actual modeling aspect here is very important as well. And in our collaboration, I mentioned that there are two facets to this. There's one where we kind of have independent modeling, where we'll model and generate the single cell transcriptomics data and combine that, but in a non-fully integrated way with the cell painting. And that's where you have this kind of independent phenomaps that you try and push together, but also the multimodal, where we're really working tightly, the computational scientists in both companies, to build and to bring these models to reality, where we're using a variety of multimodal embedding tools. There are a lot of these out there, I won't dwell on that, to build these phenotranscriptome maps. And we really already begin to see the power of some of these. So it's really exciting. We're really excited to build on this collaboration as we move forward. This is important. The models are only as good as the data. It's something that we just heard from Najat as well. There are challenges here. And I just want to emphasize, from my perspective, what I see as four of the major ones, and I guess comfortingly, a lot of it overlaps. Data management, metadata, and access. There's a lot of data out there. I think historically one of the challenges that you will have in a large pharma is that maybe that infrastructure, that data layer, has not been as coherently set up as it could be. We see that, and that ability to really build that is something we're emphasizing heavily at the moment, to make that data accessible, and especially in a forward-looking way. Because, and a statistic that I always like is that in the next 2 years, we'll generate more data than the field has in the last 40. So we should be forward-looking here. We have to be thinking about how we grandfather over those valuable data sets, but we have to always look forward, because that's how we're going to push this. Integrating expertise from multiple disciplines. We've heard about the importance of these different disciplines. We also heard a little bit about the culture. That's very, very, very important, that these different groups feel comfortable talking to one another, that they feel comfortable working together, and that we do begin to understand and speak the same language. There has been a little bit of a history. I remember when I started my PhD 20 odd years ago, so quite some time in the distant past, there was definitely a sense that computational biologists were a service group. You would throw data to them, they'd generate something, it would be passed back over the fence, and someone with an understanding of the biological system would do something interesting with it. We've got to get over that. That's not where we need to be. We need to be fully engaged with one another right across these disciplines. If we don't do that, we're not going to take advantage of these tools properly. we're not going to be able to really grasp their full potential. I think that's still a challenge. If you talk to colleagues in other industries, they would say that 80% of this is a people problem. Really getting them to use the tools properly, getting them to use, we heard about Lou, right? That's the name of your system. You've got to not use it like Google. You've got to actually talk to it and engage in a conversation with it. And for many people, that's a very different way of thinking. We're learning a little bit with ChatGPT, other bots, how to engage like that. It's still not easy. It's still very much the tendency for many people to use this rather like a Google search engine. We're not going to get the most out of it. So we have to train, we have to learn, and we have to be respectful on all sides that all of these expertises, the basic science, the computational science, the infrastructure, and that collaboration externally is going to be necessary for this to succeed. Scientific computing, again, I think this is something that we would share, that need to access outstanding scientific compute is going to be absolutely essential. And maybe different models, maybe we need to have more bursty access to compute rather than access to just large volumes. And also there's access to the really very, very good software engineers that are able to optimize the code, that are able to make it run fast. These are difficult people to find. They're, of course, in huge demand across a number of industries. But if we are able to grasp that and to really exploit that and to work collaboratively, we do this in a part of our partnership with NVIDIA, that's how we're going to accelerate the models. And finally, democratizing access and ensuring use of the data and the models. And here I think that we look very excitedly towards autonomous agents, agent-based models as a way of being able to really democratize that access, where you have that garden of software, that garden of data sets, and the agents are able to reason about where to go, and they're able to engage with the users from all disciplines, not just those that have a computational background, to really dig into that data and to make the most of it. However, this is all worth it. It's hard, but it's going to be worth it, because I really believe that the impact of this, if we get it right, is going to be transformative. Again, transformative medicines is what we are, because transformative medicines change people's lives. At Genentech, we're very happy to work with all of you guys here. It's been a great partnership so far. I look forward to many more years of fruitfully collaborating. Thank you for the invitation. I look forward to the rest of the day. Thank you.
Spoken like a true recursionaut. Actually, John and I were joking earlier today when we were flipping through the slides, whether we should take out the slides that feel redundant. And it feels like the presentation would have gotten compressed quite significantly. I hope it's striking to everybody how similar the vision is across these two organizations. And I'll just say, like, on behalf of the Recursion team, like, it's a real privilege to work with you and your team. I think the vocabulary that we use, the way we think about problems is so, like, well-tuned with one another. And so again, we're looking forward to sharing more updates about the Roche Genentech collaboration in the near term. Thanks again, John. Turning a page, so in September of 2020, as many of you know, we entered into a multi-year collaboration with Bayer. And in November of 2023, just this past year, we transitioned that collaboration moving forward to focus on 7 projects of relevance to undruggable oncology targets. That work around this new reshaped oncology interest began in earnest in Q1 of this year. Here we stand today, here in June, the end of Q2, and I'm excited to announce that we've recently initiated the first joint project under this collaboration, which is now moving quickly towards lead series nomination. Additionally, we are well on track to deliver 25 multimodal data packages in Q3 of this year that will provide a lot more substrate for additional project nominations here in the near term as well. As you already heard about today, and I want to re-emphasize, we're really excited today, this morning with our 8K, and throughout the day today, you've heard that Bayer will also be the first beta user of our low LLM orchestrated workflow engine software. Low is going to get rolled out across the collaboration, and as I think you probably picked up on if you went to the demo, it offers a lot more of an intuitive and exploratory research environment for scientists on both sides of the partnership. So extremely excited to make that announcement today, and very excited to see where that goes with our collaborators at Bayer. Earlier this year, we had the privilege of hosting many of our Bayer colleagues here at our Salt Lake City headquarters I think as many people in the audience can appreciate, it's a different feeling when you can actually walk around, see the robots, and the scale of data generation that we're able to produce here at Recursion. But that's just my words. I really want you to hear firsthand from our buyer counterparts as to their reflections on the day.
Today was an incredibly exciting day for us. We got an opportunity to host our buyer partners for the very first time.
This is a collaboration that has been ongoing for quite some time, but this aspect of the collaboration is new.
We're going to be able to explore a really large number of targets for oncology, which is something that traditionally we're not able to do.
We've made a lot of strides in cancer treatments over the past few decades. There have been several revolutions around immunotherapy, targeted therapy, angiogenesis. And looking beyond the lamppost, looking beyond the known biology is what Recursion's platform promises to unlock.
It's a new way of thinking about how to do target discovery in precision oncology.
The problem is really the on-target druggability for various reasons. So what I'm really looking forward is to explore novel biology associated with that target space.
It's really allowing us to do unbiased discovery.
Artificial intelligence relies on a huge volume of data, and that data is being generated impressively fast and at scale here at Recursion.
Obviously the automation is exceptionally cool, but it's the scale with which the automation is occurring. These are experiments that I think all of us understand how to do in very small ways, but seeing the scale with which you can do that and generate all of the data was really remarkable.
This partnership offers the opportunity, the best of both worlds, bringing the real technology forward and artificial intelligence leading edge emphasis of Recursion with the legacy of drug development from Bayer.
It's always easier when you let the partners do the talking for you. So in addition to our industry-leading collaborations with pharmaceutical companies, we have and we will continue to partner with leading companies in the technology and the data ecosystem as well. Reference has been made throughout the day to our invaluable collaborations with Tempus and with NVIDIA. Additionally, we have formed a very deep relationship with our partners over at Enamine, a Ukraine-based chemical synthesis CRO. We've brought our software tools, many of which you've heard about today, to the Enamine collaboration, and collectively we've been able to more efficiently explore this massive chemical universe that you've been hearing about. And also we're working towards developing novel compound libraries, both for Recursion's benefit, but also for Enamine customers' benefit as well. Additionally, I want to highlight one agreement that we entered into with Helix just a few weeks ago in May of 2024. Helix is a leading population genomics company that's enabling health system partners, life science organizations, and payers to accelerate the integration of genomic data into both patient care and therapeutic development. Well, I'm really excited about it. Helix is building this sustainable model of generating clinical genomics data by pairing Helix's whole exome sequencing platform with standardized EHR data across a really diverse network of health system partners. They're shown here on this slide. Representative examples of those health system partners include St. Luke's Health, WellSpan, and many more. Collectively, these entities have committed to contributing 1 million clinical genomic records over the next 3 years, which make this an extremely rich source of data moving forward. At Recursion, and under the terms of our agreement, we have access to hundreds of thousands of these unique de-identified clinical genomic records every single year. And we look forward to using that data in order to train more causal AI models like you heard about this morning, to design biomarker strategies, patient stratification strategies, and ultimately identify novel targets and medicines to impact human health. So in wrapping up, these three this three-prong approach is really driving value continuously at Recursion, with leading collaborations across pharma and the technology ecosystem. We believe that we are incredibly well prepared to accelerate our mission to impact human health at scale, and I very much look forward to sharing more updates about all of our collaborations in the near term. Thanks so much.